Efforts are underway to address structural racism in medicine, including in oncology, but cancer research and care practices that adversely affect people with the Duffy Null phenotype are an under-recognized and widespread source of discrimination. I'm Stephen Morrissey, managing editor of the New England Journal of Medicine, and I'm talking with Andrew Hantel, an assistant professor of medicine at Harvard Medical School and a faculty member in the divisions of leukemia and population sciences at Dana-Farber Cancer Institute and at the HMS Center for Bioethics. Dr. Hantel has co-authored a perspective article about the Duffy Null phenotype and cancer. Dr. Hantel, how common is the Duffy Null phenotype in the United States, including among members of various racial and ethnic groups?
So the Duffy Null phenotype is seen in approximately 2/3 or 67% of people who identify as African American. It is also present in a number of people who identify with ancestry from Arabia, as well as some individuals from different portions of South America.
You say in your article that this phenotype is associated with a circulating absolute neutrophil count that's about 40% lower than that in people without the phenotype. So what explains that association, and how does a low neutrophil count affect the health of this population?
So there is not a definitive answer as to why people with the Duffy Null phenotype have a 40% lower circulating absolute neutrophil count. It is thought that there are differences in chemokines and chemotaxis associated with it that may preferentially have neutrophils located in other tissues outside of the bloodstream, such as the spleen or the bone marrow, but we don't know for certain. Nonetheless, we do know that there are not associations between this 40% lower circulating neutrophil count and increased risk of infection in those individuals. It actually is protective against malaria, or at least two subtypes of malaria, because having the Duffy Null phenotype stops the malaria parasites from entering red blood cells, even though the white blood cells actually don't have any expression of this Duffy antigen, this Duffy blood group, which is on the red blood cells. It does have some of this chemotaxic effect, but the extent to which it actually causes any differences in infection is nil.
So despite that, although the variant is common and its effect on neutrophil counts is predictable, you say in your article that cancer research and care practices continue to discriminate against people with this Duffy null associated neutrophil count. So, for example, how have clinical trial eligibility criteria excluded some people with this phenotype?
Clinical trials have eligibility criteria in order to select the population as well as to ensure to a reasonable extent the safety of people who are enrolled. And they use absolute neutrophil criteria quite frequently because a lot of the medications used in cancer care, both the approved medications and the experimental ones, have some expectation of increased infectious risk for the population. And while neutrophils are not a perfect analog for predicting infectious risk, they are a very ready and accessible way to kind of identify individuals who may be at increased risk for infection. And so frequently, chemotherapeutic trials include ANC or neutrophil-based criteria in order to exclude people who may already be at very high risk for infections, which could be accentuated by delivering chemotherapy to them. And so the issue for people with Duffy Null phenotype is that these criteria have been agnostic to Duffy Null, even though, as you said, there's about a 40% difference between the circulating neutrophil counts of people who are Duffy Null and Duffy non-Null. And on top of that, the counts themselves, both the reference ranges we often use in clinic and the specific eligibility criteria, are largely based on reference ranges for this Northern European population, which means that they are often set too high for people with Duffy Null phenotype. For instance, for many trials, a very common cutoff for neutrophils is 1,500 cells per microliter, whereas the reference range for individuals with Duffy Null in the United States, the lower end of that is 1,200. And so you would, by nature, you're saying you're cutting off kind of a good proportion of people with Duffy Null just by making that criteria too high in their reference range.
And then another area of discrimination is adverse event grading, particularly the classification of neutropenia. So why are current classifications problematic, and what options exist for revising them?
So the current adverse event criteria, which is defined by the common terminology criteria for adverse events by the National Cancer Institute, classifies neutropenia into 4 different grades. Commonly in cancer clinical trials, grade 1 and 2 adverse events are reported, but they generally don't require changes in people's therapy, whereas grade 3 or 4 adverse events usually require changes on therapy as part of the clinical trial. And in general, all of the grades are reported. So I would say there are 2 different issues that come out of that. One is that currently grades 1 and 2 neutropenia actually fall within the reference range for people with Duffy Null phenotype. So this means we are saying that at people's healthy baseline, we are calling them to have an adverse event, which I just think is inappropriate. Moreover, for people who start off at that grade 1 or 2 baseline because of Duffy Null, not because there's actually any adverse event going on, they're much more likely to progress into a grade 3 or 4 adverse event when they're getting treatment, if it lowers their neutrophil counts even just a little bit. And this leads often to changes in that person's chemotherapy dosing, and it often makes changes in the drug label or consideration of dose modifications for individuals after a drug is approved, because as oncologists, we often look at the drug labels, the experience of the trial to help us understand where we should make modifications even after FDA approval. And we know that this is important because if people receive less than about 85% of their intended dose intensity for a chemotherapy regimen, and again, this is in general, we know that there are shortened survival, that they have shortened survival after receiving that lower dose intensity. So we think both in terms of the grading scheme itself and in terms of its effect on patients' health, that there are issues in both of those.
In addition, current criteria for remission, you say, may be inappropriate for patients with Duffy Null associated neutrophil count and hematologic cancers. So why is that the case, and how could inappropriate criteria affect patients?
Currently, multiple of the hematologic malignancies have remission criteria that include neutrophil counts, but like we've said for these other problems, they are agnostic to somebody's Duffy status. And in one case at the moment, CLL has a definition of complete remission that includes a neutrophil count that is within the Duffy Null reference range, meaning that there are some patients with Duffy Null who may have CLL and may be in remission, but their neutrophil count is not high enough to actually qualify for complete remission by the current definition. In other cases, the neutrophil counts and their thresholds for remission are lower. So in general, we would expect people with Duffy Null to also be able to meet kind of those criteria, though at the same time, in general, with this being a kind of 40% less circulating neutrophil count, we think that these people are likely to have prolonged neutropenia relative to others. And in some cases, it may lead to longer hospitalizations. For instance, the other day I was in the hospital waiting for one of our patients' neutrophils to recover after leukemia treatment. They did not have Duffy Null, but at the same time, if we were waiting around, their counts would have likely been 40% less had they had Duffy Null, and they may have had to be there for several days longer before we were able to pull back on antibiotics and allow them to leave. So in general, it's this idea that all of these criteria, these scores, these adverse events are agnostic to Duffy Null, despite us knowing that there's this significant difference between people who are Duffy Null and non-null, and kind of treating people with reference ranges that are inappropriate for them.
Nonetheless, you say in your article that you don't support race-based modifications to address disparities associated with the Duffy Null phenotype. Why are those sorts of approaches not appropriate?
So, as I mentioned with the first question, 67% of African Americans in the United States have Duffy Null. And that means that around 1/3 are Duffy non-null. And so having a cutoff that is purely race-based would kind of be appropriate maybe for 2/3 of African Americans, but inappropriate for 1/3. And for that 1/3, we would potentially be exposing them to unnecessary risk by allowing their neutrophils to go too low, even though that's not their actual blood group, that's not their actual risk profile. And at the same time, there are other individuals who may not self-identify as a group that we know to have higher Duffy prevalence, but may have genetic ancestry that actually means they do have Duffy Null. And we need to be able to treat people as individuals with risk and our ability to understand that risk that is appropriate for them and not just based on these social constructs. And so we would support individual Duffy testing to allow us to identify the people in whom we should be able to have these Duffy specific categories align with their risks of neutropenia and allow us to deliver chemotherapy when we should, pull it back when we shouldn't, allow them to participate in trials when it's safe for them, and exclude them from trials where they may be unsafe participating.
Finally, what steps can individual investigators, individual clinicians take to help ensure that people with the Duffy-Null phenotype receive equitable treatment when it comes both to research participation and to clinical care?
So, in terms of clinical trial eligibility criteria, I think in general, our trials have been overly restrictive and tried to create a homogeneous population that is often too different from the real world. And so, in general, I think having neutrophil criteria when they are appropriate based on anticipated drug safety, but not having those criteria when they are not. For when they are needed, I would say eligibility criteria for neutrophils should be Duffy specific. And so there should be screening for Duffy, and that people who have Duffy null phenotype should have potentially a different cutoff than people who are Duffy non-null, because as I said, the risk for them having neutropenia in terms of a single cutoff is very different. And in general, for people with Duffy null, ANC or neutrophil cutoffs should be really reduced to below the lower limit of their reference range, which is around 1,200 neutrophils per microliter. For the adverse events, I think we should modify those criteria, the grades 1 and 2, to specify that they don't apply to people with Duffy null, whose neutrophils are often in those ranges as their healthy baseline. I think for the adverse events, the clinicians should really look to work with the NCI and the FDA to really change those and establish more expert consensus for Duffy null associated adverse events in that realm. From the dose modification standpoint, I think more information is needed about what the specific dose modifications should be for individuals with Duffy null. I think right now it is difficult to say for the practicing clinician whether or not they should lower the dose modifications for individuals they find to be Duffy null, at least for the neutrophil related dose modifications, because we don't know exactly where that safe level of dose modification is. And so that would require further research before we can make changes. But lastly, remission criteria, I think those are also kind of standardized right now. And the main way clinicians should take part in kind of rectifying this issue is to go to the individuals who help define the remission criteria in their respective diseases and kind of pose the question to them about how and if it should be accounted for and what evidence is needed to be generated if they don't think any changes are warranted at present.
Thank you, Dr. Hantel.